Okay, parts of the microscope. So here we've got the stage. That's the flat part where you put your microscope slide in to look at it. The thing that spins around with all the different magnifications. Remember, these are called objective lenses. Those are objectives. All right, and then at the very top, you have your eyepiece. You can call it an ocular, whatever you want. At the very bottom, we have the base. Wherever the light comes from, just call it the light source or light, whatever you want to call it. On the side, the large dial, part of the dial is the coarse adjuster. If I adjust here, I'm using the fine adjustment. So coarse, fine. Very top is just called the arm. Pretty easy there. Underneath, this little lever that spins back and forth, that's the diaphragm. Or worse, you might call it the condenser, something like that. Aside from that, those are the main parts of the microscope you need to know. Just know arm, base, coarse and fine adjustment, stage, objective lenses, light source. That's pretty much everything you need to know, at least for my purposes. Alright, so animal cell now, it's pretty much the same layout. The nucleus is all the same. Nuclear envelope clears the nucleus because it has the DNA in there. The orange in the center, that's the nucleolus. Got the mitochondria, it's the same. Golgi apparatus stacked like nice pancakes. It's launching out some proteins or some other packaged material, that's the same. The rough and smooth ER, those are the same. There's really just three big differences here from the plant cell. In the animal cell, we have these centrioles. For my purposes, all you need to know is that is going to help move around chromosomes and things like that. Now, the thing that a lot of people get confused on this model is what are the vacuoles and what are the lysosomes? How do you tell the difference? So, this is for my purposes now. We're going to call these clear, these yellow, clear, the vacuoles. Why? Because vacuoles are storing things like water. Sometimes they'll store food or things like that, but for me, know that they store water. Water's clear. So the clear structure, the yellow on the outside, clear on the inside, that's going to be our vacuole. The lysosomes, because they're involved with digestion, breaking stuff down, they have these acid or bile or some other kind of potion that they use to break down molecules. So that's why the lysosome is going to be the black. So if you see it on the inside, it's the gray. So it's gray with black on the inside, and that's the vacuole. Those are the big differences there. I'm sure you all know the outside is the plasma membrane, not a cell wall, plasma membrane. Alright, quick review of these models for your final exam, just so you know what's what, so you know what I want you to say. So we'll just start with all the green are chloroplasts. So this is a chloroplast. These have been chopped in half just to show what's inside, how they're all stacked up nicely. The other whole green structures are chloroplasts that are still whole. Chloroplast, obviously, it takes in the light energy and makes food for the cell. So the orange here is the mitochondria. This one's been chopped in half. There's orange here and here. Those are ones that haven't been chopped in half. You know it's mitochondria because there are folds here on the inside. And that's that cristae, the mitochondria. Everybody knows mitochondria makes energy for the cell. And that's all I want to hear. This pink structure here, this is the Golgi apparatus. It's stacked kind of like pancakes. And so that's going to package and ship out proteins, other large molecules, things like that. And that's what these pink are here. These are proteins or some other molecule that's been packaged by the Golgi and launched out of the cell. It's getting ready to get thrown out of the cell and go to other cells or something like that. Alright, so this whole nuclear unit, let's break it down a little bit. The very outside, the white, is the nuclear envelope. On the inside, the clear is actually the nucleus. You know it's the nucleus because if you look inside, you'll be able to tell this in person better, but there's actually rolled up DNA in there. And the orange part is the nucleolus, that small structure inside the cell. Alright, so continuing on, this is the endoplasmic reticulum, the part where it's got the light green and the white dots, that's the rough, that's mainly going to make proteins, and the smooth is the blue, and it's mainly going to make lipids and things along those lines. And then everybody knows on the outside, cell wall. This is not a plasma membrane, it's a cell wall. It's much more rigid and gives the shape of the cell, gives protection for it.